Thanks, Jonathan. Let me add my welcome to all of you. It's great to see many familiar faces back here in the building, and also welcome to everyone joining online. It gives me great pleasure to introduce our first speaker for the day. We have two in-the-room presentations and one online presentations for the next hour and a half. I think it's always great to get the Cheminformatics community together on a quarterly basis here, so let's have another great session today. The first speaker will be Layla Hosseini-Gerami from Ignota Labs. And she'll be talking about a day in the life of a chief data science officer making data-driven decisions in drug discovery. So Dr. Layla Hosseini-Gerami leads the data science efforts at Ignota Labs, where she applies AI to uncover and prevent drug toxicity. With a strong foundation in chemistry and a PhD from the University of Cambridge, her work bridges molecular science and machine learning. Her research has led to new insights into how drugs interact with the body. Now she focuses on turning those insights into safer therapies through innovative biology-aware AI approaches. It gives me great pleasure to welcome Layla.
The wrong one. No worries.
That's you. Yep. Super.
Perfect. Thank you very much. Can everyone hear me okay? Cool. Thank you for that introduction, and it's really great to be here. So my name is Layla. I'm going to be talking a bit about the day in my life as a Chief Data Science Officer. I'm going to start with a few slides about kind of what we do as a company. We're pretty new, so you might not have heard of us, just to give a little bit of context to the day. Oh, wrong one. So why do we exist? So 2 billion patients are waiting for suitable treatments for their condition, but promising candidates are abandoned early, and we are here to change that. So putting that into numbers, out of all of the drugs that enter regulatory preclinical trials, up to 56% of them will fail due to safety issues, usually at the preclinical or phase 1 phase. This amounts to over $400 billion lost every year. But why do these drugs have to fail? Well, for the R&D scientists, they might get an output that the drug has caused liver damage or heart attacks in patients. That output is useful to know that something has gone wrong, but it doesn't really tell you how to fix it. For the biotech executive, safety failures cost them billions, but fixing them is a much higher risk compared to progressing a new drug and starting again. Ultimately, that means for patients that they don't have the medication they need to improve their health. So this is where we come in to revive failed projects, giving fresh hope to scientists, investors, and patients. So we do this in a kind of 3-phase process. The first phase is to identify. So this is targeted lead validation to find these failed drugs and identify potential deals at scale. The second phase is to fix them, using our platform to identify new chemistry and remove safety issues. And then the third part is developing, validating the new chemistry and getting it back to the clinic as fast as possible. So I'm just going to quickly go over the first 2 buckets and kind of how we do them. So the first one is, how do we identify? So we've built a tool called Radar, which is an asset finder powered by large language models, where we take things like industry news, clinical trial data from clinicaltrials.gov or global data, as well as network leads through conferences that we attend. We then use an agentic researcher to go and do some research on that drug. So obtain information about the chemical structure, the patents, the commercial potential, its target, and where else it can be used. The information is summarized and kind of generated into a report that a scientist can read. And it's also then passed through a fact checker, because we know a lot of the time, if the language model doesn't know the answer, it will make something up. And the output is a comprehensive report for us to prioritize interesting opportunities. This is just an example of the kind of thing that comes out of Radar. So you can see we have an executive summary. We have kind of censored the targets and the compound names, but you can see that we get a lot of comprehensive information out there that really helps us to make a decision. Then moving on to how do we fix? So we've developed a platform that we call SafePath, which is essentially a combination of cheminformatics and bioinformatics. So using cheminformatics to understand what are the potential off targets, and then using bioinformatics to understand what does that mean in terms of the downstream effects that might be happening with that drug. So this doesn't work so well with the downward scrolling, but this is essentially a schematic of how it works. So we first map all possible target interactions across biology, understand the subsequent cause and effect cascade from target to toxicity, and this allows us to do things like differentiate between species biology. If we know what went wrong, perhaps in a rat, can we see whether that would happen in a human, and vice versa. So we start with a compound structure and we have an output which we know, which is, for example, the drug has caused liver toxicity in rats. We have a large knowledge graph, which contains lots of information connecting different data modalities together, and it's just too large for a human being to traverse by themselves. But what we can do is we can map a path from that compound structure to that toxicity outcome, and then really understand on a granular level exactly what's happening. So in this case, a toxic metabolite is being generated via CYP2D, which is inhibiting the bile salt export pump. And then we know through the knowledge graph what this leads to, which is bile acid accumulation, eventually leading to cholestasis and liver toxicity. So this gives us these really granular and testable hypotheses. And with the inclusion of cheminformatics, we can even drill into what part of the chemical structure is driving that unintended interaction. So why is that compound being metabolized via CYP2D? Why is it inhibiting the bile salt export pump? And then that can tell us what can we do to change it. So that was kind of in a nutshell what the company does and what we do. So now I'm going to move on to a day in the life. So my role at Ignota Labs is as Chief Data Science Officer, which means turning very complex data into clear decisions and making sure that our science moves just as fast as our ambition. So no 2 days are ever the same, but most of them follow the same kind of rhythm, which is balancing decisions, data, and people. So here is how a typical day goes. 9 a.m. So starting the day is always kind of checking up on what's happened overnight, right? So looking at emails, looking at our Slack messages for anything that's popped up overnight. Maybe we've been running some models overnight that we want to check up on in the morning. Maybe some team members have sent messages or emails from partners that we need to action. So at this stage, we can start to flag kind of early wins or some red flags. Maybe a run failed and we know, okay, right, I need to look into this at some point today. Maybe something will come up that changes our priorities. So this is really the kind of start of the day and sets us going in terms of, yeah, what we're going to be looking at that day. So at 9:30, we have our stand-up. So we're quite a small company. There's only 6 of us. So we're lucky enough that we can do a whole team stand-up where everyone can kind of talk about what they're up to. So within this whole company sync, it's an opportunity for all of us to understand who's doing what and why. So this is not only the technical team, but also the business development side of things, the scientific development side of things, the fundraising, and it really gives everyone a good picture of what's going on within the company, even if it's not within their specific remit, because we're, as I said, small company, all working towards the same goal. It allows us to surface any blockers early. Maybe someone's waiting for me to finish a piece of analysis, and without that, they can't continue. So it's a way for us to say, I need to do this, but I can't do that until you do that piece of work for me. Also allows us to redirect if someone's stuck or they're solving the wrong problem. And it just allows us to kind of clarify our focus for the day. So maybe I could say, guys, I checked and that run last night failed, something's gone wrong, and we need to have a look at that today, when that wasn't necessarily what we thought we'd be looking at today. So 10 a.m., catching up on papers. This is a really crucial part of the role. Staying sharp on the AI frontier. So we all know that there's loads of papers coming out all of the time, and it's kind of a job in itself to keep up with all of it. So this is why it's important to kind of have the time set out in the day to be able to look into these new papers that have come out. So they might be new model architectures, new applications in drug discovery, sometimes an interesting paper that has nothing to do with drug discovery, but we can kind of see that there might be an interesting application within there. We focus on what's actionable. So what is out there that could impact how we build, how we interpret, or how we prioritize. Then I kind of save those kind of standout ideas, the ones that I really think will make an impact, to bring into upcoming tech team syncs. And the kind of places that we tend to find them is Archive, Twitter, or more recently Blue Sky, Papers with Code, and those kind of sources. So for example, I might read through and see, oh, this is a great paper, could be a really great way to split our drug target model data sets. Biochem Insight, looking at recognition of optical chemical structures. This is great. We can use this to improve our pattern extraction process in radar. So really looking at ways that we can pick out the latest advancements in AI and how we can build on them within the company. So 11 a.m. is then the tech team update. So I mentioned that we have a morning stand-up where everyone in the team talks about what they're up to. This is kind of a high-level overview. It's just giving a kind of basic understanding of what everyone's up to. So we have weekly separate tech team syncs where the people actually working on the tech and on the machine learning can get together and talk in a lot more detail, a lot more granularity, kind of the stuff that, for example, the CEO doesn't really care about. He wants to know why we're doing things and the progress of the larger goals, doesn't necessarily care about things like, you know, oh, there's imbalance in this data set that we need to fix, for example. So this technical deep dive, as I mentioned, it's a weekly touch point with the engineering and machine learning teams. We can review progress on things like our model building, anything that's blocking us. We discuss things like technical trade-offs, speed versus accuracy, how are we going to scale? We're at a stage where we're growing quite rapidly. So that's something that's in particular focus for us right now. Also bringing in any relevant insights. So saying, guys, I read this really great paper. I think we should have a look at this in more detail. And then really deciding what gets prioritized in this sprint and what needs revisiting. And what I mean by sprint, because this is something that I don't know if everyone is familiar with, but it's essentially a way of how we organize our work and our priorities. So we have 2-week sprints, where at the start of the sprint, we set our goals, we say what we want to achieve in the next 2 weeks, making sure this aligns with the kind of wider company goals. And then as we go through, we know exactly what we need to do in that time. So I'll give an example. I call it one board to rule them all. I haven't put our actual sprint board up here because there's some like sensitive stuff on there, but we use Notion and this is a really great tool. And you can see how you can kind of organize tasks into different projects. You can assign priorities, you can assign people to the different tasks. And this just means that we can keep things as organized as possible when we're kind of working out what to do and kind of the order in which to do them. 12 o'clock. This is a very important part of the day, lunch and reset. Now, if you talk to a lot of founders, they'll probably tell you, I don't take lunch breaks, I eat lunch at my desk, you know, stuff like that. I don't believe in that at all. So lunch is a really important time for you to kind of take a little break. You've been thinking hard since the morning, and I try to go out for a walk, clear my head a little bit. And I tend to find that the most useful ideas come up when you're not trying to force them. So I might go for a walk or go for a run or whatever it might be, and an idea will pop into my head, or maybe suddenly a solution to a problem will pop into my head. And it's just a time to kind of step away a little bit. And as I say, I think that's really important. And I think it's something that I think a lot of founders would say, oh no, I don't do that, trying to look like they're really hustling and really working hard, but I actually think that's quite detrimental. Right, back from lunch, it's 1 o'clock, and now we're having a sync up to talk about different asset opportunities that we've seen. So in these sessions, we ask, what do we have and what do we need? So as I mentioned, we have this tool where we can identify interesting opportunities in terms of assets to in-license and fix. So we can review them, what's new, what's resurfaced, what's interesting, both scientifically and also commercially. And then most importantly, we ask the question, do we have enough data to be able to evaluate this property properly and be able to make a decision? What do we need in terms of technical progress to actually validate this? Sometimes what happens is we see that one asset has stood out with really amazing potential. We have early data that looks promising, but there's some gaps that we need to fill, but we might need to move fast. We might say, okay, we found this and we need to move super fast. At this point, we need to make a decision that this jumps the queue. So we've had our morning stand up, we've said this is what we're going to do, we've planned out our sprint, but inevitably something will come up at some point where we kind of have to reprioritize, drop what we're doing and focus on something else. So 2 o'clock, we've just decided something urgent has come up. We need to reprioritize and speak to the team and kind of convey this information to them. So at this point, we need to shift gears and go. We've had our nice relaxing lunch, so we've got plenty of energy to go and do it. So at this point, might need to block off some time to develop a model very quickly or do a feasibility analysis, telling the team we're shifting the focus and this is why, and that's really important. Again, that kind of communication piece, kind of telling them we're doing this now. Don't ask any questions is not the right way to do it. So really being clear and saying, this amazing opportunity has come up. We need to get back to them yesterday, so we need to do this right now. Dropping the other threads for now, because we have Notion, we can go back in and we can deprioritize tasks and add new tasks and just make it very clear what we're working on. You might say that at that point, the goal is to unblock the downstream team, so the scientific development team and the drug discovery team by the end of the day or the first thing tomorrow. 3 o'clock rolls around and it's time for a meeting with a collaborator. So we work with different partners through different means. So right now we've got an Innovate UK grant working with a company called Cytochroma who are doing some screening for us. So meeting with them to review any new data. So they're actually measuring 2 types of data for us, which is cell painting and gene expression data for a toxic drug on a liver cell line. So at that point, we'll look together, look at how the compounds behave. Are there any unexpected phenotypes or signatures or correlations? Is this telling us something or is it just noise? So really having that conversation with a collaborator and really understanding that data and what that means. So we have our new data set, and now it's time to design the next phase of analysis. So at this point, we start making plans. So reviewing the notes and the outputs from the collaborator meeting, we have some of the data in hand, defining the questions we need to answer, looking at, for example, which phenotypes are unique to this compound, do these changes in transcription or gene expression match any known compounds with known toxicities, drafting the analysis plan, looking at what methods do we want to use, what's our baseline, what are our controls. And then we come to the end of the day where we wrap up. So the end of the day is a very important time because it's very easy to kind of just close your laptop and go off, which is what we all want to do. But at this point, it's things like capturing the key decisions from the day. So as I mentioned earlier, we had this really big opportunity come up that we had to go and chase, kind of writing that down and capturing. This is one of the decisions that we made. We decided to stop working on X to start working on Y. Flagging anything unexpected, perhaps in that analysis I did with the collaborators' data, something looked a bit funny that needs a bit more analysis. I need to kind of make a note of that so I don't forget to come back to it. And also things like queuing up stuff that can run overnight. So say, for example, there's a big model training job that we want to do. I know that if I set it off now, I can come back in the morning and check it then. So to summarize, what does this role really look like? There's a lot of context switching, kind of changing between like the strategy side, the science side, and the tech side. And my role is to really kind of bridge all of those things together. As I said, we're a small, agile company. Fast and informed decisions are sometimes more important than making the perfect decision. Sometimes you have to make a decision on limited data or by making assumptions. Translating between what's possible, what's valuable. So sometimes we have a bit of a pool. Maybe the scientific team would say, it's really good if we could have this so that we can help with our drug development. The tech team will turn around and say, you know, that's not as quick of a project as you think it's going to be, kind of saying, oh yeah, just build this thing for us. You know, can you have it by next week? So kind of also doing that translation piece of, you know, this is what we need and this is what's actually possible, and kind of bringing the realism a little bit sometimes, and then clearing paths so that teams can move without friction. And that's it. So that was, yeah, an intro to the company and just a kind of typical day in the life for me. So thank you very much for listening.
I think we have a bit of time for questions, I would have thought. So we've got a microphone for the room, and also people online. You might want to put things into the chat box. Any questions? Basil.
Hi, Layla. Thank you. I enjoyed that. Can you tell us a bit more about how you capture?
One second, we'll take a microphone. 1 second.
People online.
People
Thanks.
online can hear
Okay.
them.
Yes, thank you, Layla. Can you tell us a bit more about how do you capture commercial value in your decision making?
Yeah, absolutely. So we do this by, first of all, looking at the indication space that the drug is in. So, for example, maybe the drug that we're looking at was originally in obesity, for example. We have a look at the target and what other opportunities there are for that in terms of other indications. And then some of the research that this tool does is kind of a market analysis. For us, it's really important to have a market to sell to afterwards, because we typically don't take on the drugs to take all the way to the end. We typically want to put them back through a preclinical and phase 1, and then at that point sell them on. So also getting information on what's the selling market, and tools like global data are really helpful for that. It is an expensive subscription, but definitely very worthwhile for us to get that information.
Thank you.
Jonathan.
Hi, Layla. I think one which maybe this tsunami of information that all of us are facing here. You talked about your daily review of the literature and incoming. I think it sounds like a really useful discipline. Is that enough though, the half hour? I mean, do you have any tools or hints for people about how to, because there's so much out there, and how do we sort of identify what's really relevant and zero in on it?
Yeah, I mean, I think first of all, like the real answer to that is like there's never enough time, and there's always stuff that's out there and always stuff that we'll miss. For me, it's about like certain keywords on Google Scholar that you can kind of subscribe to. So things like, I don't know, molecular property prediction, even things like new toxicity databases, just different keywords that you can put in. And then I tend to find that that gives a nice summary every day of new articles that have come up that could be relevant based on the keywords that I'm interested in. But there definitely isn't enough time in the day to look at all of it. It's about kind of I've kind of gotten good over time at being able to skim abstracts quite quickly and just quickly decide whether, yeah, I'm going to keep reading, or no, I'm going to move on to the next one. So also being a bit quite picky on there might be something that I'm like, this is really interesting, I'd love to read it, but it's not very relevant. I'm going to read that in my own time. So
You haven't got an AI on top of an AI yet to scan that for you.
I mean, you can. So sometimes we do put papers into even like ChatGPT. You can upload the PDF of the paper and just say, give me a quick summary. And sometimes that's quite helpful. Maybe when you've got quite a few that you want to review, but you don't have enough time to read the whole paper, or like read all of the papers at that time. So getting a quick summary can also be useful, because then it can pull out the key bits of information that you actually need.
Thank you.
Good. I think we've got time for a few more questions. There's a couple online first, then I'll go back to the room, Peter, if that's okay. I'll do the online ones first because there's a couple. At one point, a graph of proteins was shown, probably related to toxicity. Where does this biological graph come from? That's a question from Francois Verger.
Yeah, that's a really good question. So we have built this graph ourselves, and it's essentially pulling from lots of different sources. I did have a slide on this, and I wish I had it as a backup. But the key relationships that we have are like drug protein relationships that we get from places like Campbell, binding database, etc. Protein interactions that we get from like Omnipath database, String, protein pathway associations from databases like Reactome or Gene Ontology. What else? Drug toxicity associations that you can get from CIDR, offsides databases. I'm probably missing them. We have quite a few. But also there's like protein side effect relationships as well. I can't remember the name of the database now. I think it's called ADREX database. So heterogeneous relationships connecting drugs, proteins, phenotypes, pathways. Yeah.
Plenty of data sources to connect for sure,
Yeah,
to
exactly.
work with. There's one more online, or two more. At what time do you check for data or references from patent literature to compare with your evidences? From Luca.
Yeah, so that is kind of part of that radar like asset sourcing process, is doing the patent scraping. So reading the patent and bringing that in as well as part of that report that we generate, which is then what the scientists within the company will review. So it kind of gets pulled in at that phase.
And then one more online, and we do one more in the room. We've got 2 more minutes here on this. From Kalen, do you reincorporate new data that is generated by your collaborator into your existing ML pipeline for a better feedback loop?
Yeah, absolutely. Yes, that's something that we do in general. When we have new data, we kind of use that to refine. So we might, for example, make predictions on a bunch of compounds. We can go and test them. The ones that we get wrong, we can then, well, also the ones that we get right, we can feed that back in. So yeah, that is, yeah, definitely something that we like to do.
Great, cool. We'll move to the room. I think there was a question from Peter, if we have a microphone for Peter. Yeah, I think it does.
I was very interested in your use of sprint, right? Because sprint is a team activity and it seems to get at the team as a whole company.
The mic isn't working, I think. Can
actually,
you use
yeah.
the other?
Oh, is it working now?
Yeah, no,
Yes,
it is.
fine. Good.
Okay. Sprint is a team activity, and here you've got the whole company acting as a team, but they've all got umpteen other jobs to do as well. So what is the focus of a sprint? Is it that you take a particular target and say, we're going to come up with, you know, a plan by the end of the 2 weeks or whatever your time scale is, and people have to partition their activity on the sprint against all the other things they're doing? Is that right?
Yeah, so we have kind of a set of company goals. So we know broadly what we want to achieve over the next year, what we want to achieve over the next 3 months, and what we want to achieve in the next month. So what we do is when we have our sprints, we kind of have a look at what are our goals, what is the highest priority, highest urgency, what do we need to get done now, and what can be done later on in the next sprint or a later sprint. And then we use that to then divide. So it might be that, you know, our CEO needs to go and find some more investment. That might also involve other members of the team. He might say, well, my goal, this is really important. We really need to get the fundraise done. I'm going to need, Layla, your time because you're going to need to speak to an investor. I need your time because you're going to need to, I don't know, write something for an investor, for example. So it's all really anchored on the goals for the company as a whole.
Great questions. I think I'll take one more, the final one online, then we move to the next speaker. One from Kepa. You have mentioned radar and SafePath. Do you create new models quite frequently? And do you also implement or use open source models?
Yeah, so it's a mixture. We do have a lot of our own proprietary models, but we also use open source things to complement. So for example, within radar, we do kind of patent scraping. So I mentioned that paper, which I'm sure we'll have a look at and implement soon. But Decima, for example, is an open source tool that's used to take pictures of chemicals and translate them into the smiles. So that is part of the pipeline within our overall kind of framework of asset evaluation, using these open source tools where we can. We don't believe in reinventing the wheel if we think that it's being done good enough, unless we think that we can add kind of an order of magnitude. And so we really focus our development on, yeah, where we think we can make the biggest impact and use open source tools for the rest.
Thank you. We're now moving to the next speaker. Thank you, Layla, again. Yeah, let's need to click on. I might need some IT support with getting back to the slides if that's possible. But I will give the intro to our next speaker, which is it's a great pleasure to introduce Ida Rahu from the University of Stockholm. So she will be talking about targeting the unknown, flagging high-risk chemicals in environmental samples without full identification using machine learning. Ida received her doctoral degree in chemistry in 2020 from the University of Tartu, where her research focused on applying sustainable chemistry principles to organic synthesis. Realizing that her toolbox for evaluating large datasets was limited, she pursued a master's degree in data science, awarded in 2023, while simultaneously working as a researcher in bioinformatics. She then pursued her career as postdoctoral fellow in computational mass spectrometry at Stockholm University. Most recently, Ida has been appointed as an assistant professor in the Department of Environmental Science at Stockholm University, where her research bridges environmental chemistry and data science, aiming to address pressing challenges related to organic contaminants. Welcome, Ida. Over to you for your presentation. We just need to probably click on the 1 second. Yeah, I'll try. This might be the main thing. Is this the one? Yeah.
Yes.
All good. Can everyone see this online? I hope so. Over to you, Ida.
Good afternoon, everyone. Thank you for the kind introduction, and many thanks to the organizers for giving me the opportunity to speak here. Today, I will introduce you to my research project, which investigates of using mass spectra to predict the biochemical activity of compounds, ultimately aiming to enhance the environmental analysis. Throughout our lives, we are exposed to a wide variety of chemicals from various sources, such as food, household cleaning product, wastewater, and these real-world samples can contain hundreds to thousands of chemicals, some of which may pose risks to human health or the environment. Nowadays, for the detection and identification of known and unknown chemicals in those complex samples, non-targeted screening, or NTS in short, combined with liquid or gas chromatography, high-resolution mass spectrometry, is considered to be one of the most comprehensive methods. This approach utilizes the generic wide-scope strategy for both sample preparation and analysis to maximize the coverage of chemical space of the samples. However, this often leads to a very large and complex data sets consisting of thousands of LC or GC HRMS features. And by features, I mean the combination of retention time and accurate mass with or without the fragmentation spectra or MS2. To give you an idea of the scale, analyzing just one example, a sample typically produces about 5 gigabytes of data. It is clear that matching this amount of spectral data with chemical structures is a major challenge, and to address this, NTS workflows have developed that typically begin by assigning a set of candidate structures to each detected feature, and this is achieved by either matching the MS2 spectra with the experimental or in silico spectra from the libraries, extracting directly structural information from the MS2 spectra and comparing it with the chemical structures from the databases, or most recently using generative models to predict the structures that could explain the obtained spectra. However, not all the candidate structures are equally plausible, and therefore, the next step is usually using the complementary analytical information, such as retention time from the chromatography or collision cross-section from ion mobility measurements, to prioritize most likely structures. And at this stage, machine learning models are frequently used to predict this empirical analytical information for each candidate structure. And if candidates' predicted result aligns well with the experimental results, this candidate is considered to be more plausible and is therefore prioritized. But even with this approach, the workflows produce multiple plausible candidates for each detected feature. To illustrate this on the visualization, meaning that only small fraction, up to 10% of the detected features are actually identified with high confidence. And to illustrate this, on the visualization of this slide, environmentally relevant chemicals from Norman Sustad database are used as an approximation of the chemical space, and the highlighted points represent chemicals that are identified with high confidence in recent environmental studies. As you can see, these account only about for 3% of the targeted chemical space. So what about the other remaining unidentified chemicals? What if one of them is highly hazardous? Given the time-consuming and complex nature of the identification process, it is reasonable to ask why we don't focus all our efforts only on the detected features that are most likely to pose risks to humans or the environment. By flagging these first and prioritizing their identification, we can make the NTS workflows more efficient and targeted from the start. To do this, we would need a tool or assistant that can guide us toward most critical features. And you can think of it like using a police dog to track down suspect by its scent. But in our case, that police dog would be an in silico tool capable of sniffing out high-risk features. There are several parameters based on which one could prioritize organic contaminants, one of which is their endocrine disrupting activity. Endocrine disruptors are a diverse group of chemicals that can interfere with any aspect of hormone action in the body and thus affect the normal functioning of endocrine system. Due to their broad and significant effects, they are a major focus in current risk assessment and management plans. To predict which chemicals might exhibit such activity, we can turn to machine learning models as our police dogs, trained to detect structural patterns associated with potential harm. You all know that to train those models, these are trained typically on known structures, often represented as molecular fingerprints. But let me remind you, while analyzing real-world samples, we don't know the structures. If we are lucky, we know the accurate mass together with MS2 spectra. But there is a workaround. The fingerprints needed for machine learning can in fact be predicted to unidentified chemicals based on the structural patterns present in their mass spectra. It is possible while analyzing real-world samples with high-resolution mass spectrometry and utilizing the capabilities of tools such as Sirius together with CSI Finger ID. This software annotates the fragmentation spectrum with a chemical formula by computing the fragmentation tree that explains the obtained spectrum. Fragmentation trees are graph-based data structures that map out the fragmentation pathways of chemicals under investigation. Each node in the tree represents the fragment's chemical formula, and edges describe the losses between the precursor and product ions. And then these trees, together with the corresponding mass spectra, can be used or are used to predict the molecular fingerprints. However, it is important to notice that these fingerprints differ from the traditional binary fingerprints. Instead of zeros and ones, each fingerprint feature is given as posterior probability, indicating the likelihood that specific structural pattern is present in the chemical. The fingerprint used in this work is from Sirius version 5. It consists approximately 4,000 features. The exact number depends on the ionization mode used during the analysis and integrates several types of molecular fingerprints, some of which are also widely known in the cheminformatics community. Now, having discussed the workaround to how to generate molecular fingerprints, let's have a look how this method, when combined with machine learning, can be used to flag unidentified, potentially high-risk chemicals while by walking through the workflow developed in this study. First of all, to develop the machine learning models, relevant toxicity data was needed. And for this purpose, TOX21 data challenge dataset, which is one of the most widely used datasets in the field of in silico toxicology, was used. Now, this dataset provides qualitative toxicity measurements given as binary labels where 0 indicates inactive and 1 indicates active chemicals across 12 different bioassays, several of which are related to endocrine disruption. Even though one of the most comprehensive datasets available, it still possesses many limitations such as data duplication, missing labels, class imbalance, and therefore the first step in overall workflow was data cleaning. First, we excluded all the chemicals undetectable during the HRMS analysis and also removed the structural details that cannot be derived from the mass spectra, such as stereochemistry. After standardizing the structural notations, we deduplicated this dataset to deal with the chemicals whose experimental results were inconsistent across the duplicated entries. We didn't use, however, the majority vote approach, but used a strategy based on the precautionary principle. So if chemical was assigned to be active in at least one measurement, it was assigned to be active also in the final clean data set to ensure that none of the hazardous chemicals were overlooked. So to evaluate the developed models under real-world conditions, HRMS data was also needed, and therefore the MS2 spectra was extracted from the databases and matched with the original dataset based on the standardized structure notations. And from this the real-life test set was formed comprising the chemicals with both toxicity and spectral information, and this data set was kept aside until the final evaluation of the models. To train the models, molecular fingerprints were needed. However, only small fraction of chemicals from the original data sets had spectral data available. So that's why we couldn't use the series to predict it. And therefore, we used a different strategy to generate them. Namely, we calculated them based on the structure notation. But it is important to notice that now we have binary fingerprints to train the models. And after that, we split this data set to two parts to train and test the models using the anti-clustering algorithm to ensure an equal fraction of active chemicals in both sets. So a small conclusion, after this thorough pre-processing pipeline, we have three different data sets: training set for training the models, intermediate test set for selecting the models that go on the final evaluation, where the performance is measured on real-life test set. To evaluate the model's performance and select those that go final evaluation, of course, we needed an appropriate evaluation metric. And in the literature, it is very often seen that many metric values are reported to the same model. However, this can lead to the confusion because based on one metric, one model seems to be superior, based on another. And that is why it is very important that the evaluation metric aligns well with the model's intended application. In our case, models had to have the high recall or true positive rate to flag majority of the active chemicals in the sample, but at the same time, minimizing the numbers of false positives was crucial to reduce the post-processing workload, and therefore we used false positive rate at 90% recall as our criterion. And the lowest false positive rates, depending on the bioassay, on intermediate test set range from 0.19 for the aryl hydrocarbon receptor endpoint to 0.69 for the estrogen receptor endpoint. And it is also worth noting that, or mentioning that, while developing the models, we used several algorithms to train the models, but only those based on the ensemble methods such as random forest or gradient boosting were represented in the final selection. So now the last step of the workflow, evaluating the models on the real-life test set were based on the fingerprints predicted by series. While this tool allows us to obtain or get the information about chemicals' properties without their identification, it still entails certain limitations, such as need for data-rich HRMS or the fact that the predicted fingerprints cannot be taken as ground truth. However, the most challenging limitation arises while considering using these probabilistic fingerprints as an input of the models trained on binary fingerprints. A standard approach to overcome this is using a threshold, so all the fingerprint features with probability higher than the threshold are set to 1 and all below set to 0. However, selecting a meaningful and consistent threshold is not straightforward and can significantly affect the performance of the model. And therefore, we tried to use a different strategy where each fingerprint feature was sampled by its probability of being 1. So, for example, in this case, the probability was 0.71, meaning that there was 71% of chance that it will be sampled as 1 and 29% of chance that it will be sampled as 0. And then the whole sampled fingerprint is used as an input of the model. And by repeating this procedure enough time, the average of the model's prediction for the same chemical becomes constant and represents the underlying distributions better. And the lowest false positive rates on real-life test set achieved by this method range from 0.25 to 0.85, meaning that using these models, we can reduce the post-processing workload in NTS workflow up to 75%. Now, after developing the models that can, in fact, flag high-risk chemicals in the samples, before we really can apply them in real NTS workflows, we have to understand their decision-making process. More specifically, we have to know, does their predictions rely on meaningful chemically meaningful structural features, or they based on just random patterns? And to investigate it a bit, we performed the Shapley additive explanation analysis, and the results for aryl hydrocarbon receptor model are shown. As all of you can see, while not all the fingerprint features deemed to be important by the model are truly meaningful, some of them, however, like these annealated rings or endomoieties, are proved to be highly relevant in this specific context. And this finding is significant as it not only helps to build trust to the model predictions, but also opens the opportunities to investigate which structural patterns and how affect the compound's biochemical activity. And to also demonstrate the sniffing capabilities of our developed models, we conducted a case study where we analyzed white water samples with NTS LC-HRMS workflow. And we applied then to all the detected features with MS2 spectra, model developed for AHR endpoint. And then this model flagged 55 chemicals as potentially active, among which was also one spike chemical. And this one spike chemical was in fact the only spike chemical that was confirmed in a toxicological experiment to be active in this specific bioassay. So building on these promising results, currently we're conducting more case studies to explore the developed models capabilities further, but at the same time, we are also developing new models that use the same approach, but integrate different fingerprints or are developed to predict other relevant parameters to flag high-risk chemicals in the samples. And before I finish, I want to thank the funding sources for supporting this job, work, and also my supervisors and colleagues at both Stockholm University and University of Tartu. And I also have a small advertisement. I am currently building my own research group, which will focus on advancing the environmental science through data-driven approaches. So, and right now I'm hiring my first PhD student. So if one of you or you know someone who might be interested in, then maybe feel free to scan the QR code to get more information or try contact me directly. And by that, I want to thank all of you for your attention.
Thank you very much, Ida. We'll take a few questions. We'll probably start online for a couple, and then we'll go to the room, and then we'll go back online. So let's see. Click on the right buttons. Wait a minute. For the last one or two. Oh. No, wait a minute. Was it? Where do we start?
Yeah, the talks 21. I think that's the first question related
Which
to the
one?
stock. Talks
Which one?
21. Kaelin's question.
Kaelin, this one. Also, how did you do your data splitting scaffold split? Is that the right one? Right question? Good.
Yes.
Oh, one before. Sorry. Oh, feel free to answer that one, then we'll go to the next one. So how do you do the data splitting and scaffold splitting?
Yes. No, I didn't do scaffold splitting. As I said, for me, it was very important that the active chemicals across the 12 different bioassays would be equally distributed during the data splitting, and therefore I used anti-clustering, and I didn't look like basically the data structures directly, but more that the active chemicals would be splitted equally.
Here was the first question from online for a classifier. Try RC, AUC or MCC. Those are the proper metrics to look for. That's the more.
Yeah, actually, of course, this was one metric that we used, but of course, we can also provided other metric values and calculated them for all the developed models.
I'll do one more from online. Did you remove the stereochemistry as part of the cleaning process before evaluating toxicity?
Yes, this was this is one of the biggest problems while developing models, because we all know that while we want to predict the toxicity, the stereochemistry is very, very important. It defines for many structures their activity. But in our case, I think it's more relevant to remove it, because we will never get this information from the high resolution mass spectra. So
Kepa mentioned the thalidomide case where
Yes.
stereochemistry is rather
Yes.
very important, but yeah.
And yeah, we rather use that this precautionary principle. So we rather want to flag more chemicals than less chemicals.
Yeah.
So
Okay. I think that'd be more of a discussion on stereochemistry, but I'm going to move to the room. Any questions in the room? Layla.
Thank you. That was a really great talk, really cool work. I had a question about, did you look into why some of the endpoints you performed better in than others? Like, did you get any understanding of why that was the case?
yeah, there are several reasons, and one of them is also the data quality of the DOCS21 data challenge dataset. For example, that not all the endpoints are, like the quality of all the endpoints are not equal.
Thank you.
More questions in the room? Still got a few minutes to take a few more questions. Otherwise, we'll go back online. I think there was one more, two more from Adelaine, because the molecules need to be MS ready. Okay, no, sorry, that was more stereochemistry discussions. Would it be possible to train a VAE with the MS signatures as the latent space?
Yes, I know that it is possible, and people have tried it and also currently try to do this. So yes, it is possible. I myself don't do it though.
Any more in the room before we move to the next presentation? Well, then let's thank Ida once more for the presentation. Now, it's a pleasure to introduce our third speaker, coming from the University of Oxford, the other place in close vicinity to Cambridge. It's always good to hear from our friends in Oxford, isn't it? So next speaker is Isak Valsson, Oxford University, speaking about narrowing the gap between machine learning scoring functions and free energy perturbation using augmented data. So Isak Valsson is a DPhil student in statistics at the University of Oxford, as part of the Oxford Protein Informatics Group, OPIG, led by Professor Charlotte Dean. Before his DPhil, he did his BSc in Applied Mathematics at the University of Iceland and an MSc in Statistics at the University of Oxford. Isak will be talking about his paper, Narrowing the Gap Between Machine Learning Scoring Functions and Free Energy Perturbation Using Augmented Data, which was published last month in Nature's Communications Chemistry. So this is an online presentation. Over to you, Isak, in a second. I just need to find the right slides. Or will you be sharing from your end? How does this work again here? Check. Let's see. I press the escape button. Right. Okay. Okay.
Does this work? Can you hear me and see my slides?
We can hear you, but we can't yet see what you're sharing. So can you somehow take control and share from your end? Because I can't find the right button at this end.
Yeah, I'll try something else. Let's see.
I will look at the mouse is.
Can you stop sharing, Jürgen? Because otherwise, I'm not quite sure that works sometimes.
I need to find where the mouse is. There's so many screens here. I'm quite on this. Where was it? Over there? How do I get it over here? Ah, there we go. Got it. Right. What? Pause sharing then, Andreas, you reckon? Stop sharing. This one. Yeah. Thank you all. Good teamwork. All right. Now over to you, Isak.
Okay, so now you can see my slides, or yes or no?
Yeah, we can see them, but interestingly enough, they come out on the TV on the side of the room, so we might have to do some further jiggery pokery.
Okay.
Everybody knows what we're doing here.
Okay.
No one knows what we're doing. Certainly not me, but I can make it out.
Yeah. Okay.
Online, it's fine. Online looks good.
Okay.
Right. Good for you guys online then.
Too many screens here. Yes, please. Keith is
Yeah,
here to the
Keith.
rescue.
There we go. Minimize it. All right, we're all good here in the room. Isak, over to you. You can start.
Okay, thank you very much. And thank you for the introduction. So I'm going to talk about some of the work that I've done on developing machine learning scoring functions. And like you said, it's been published in Nature's Communications Chemistry. So please check out the paper if you're interested. So I'm going to start by telling you about sort of two different scenarios where machine learning scoring functions can be used, hit identification and lead optimization. And I'm going to try to highlight the differences between those two scenarios, including sort of what aspects of binding affinity prediction are most important in each case, and what kind of benchmarks you want to use in each case as well. Then I'm going to talk about some of the work that I've done, including developing a new benchmark called OOD test, or out of distribution test, and a new model called AEV-PLIG, and how we used augmented data to improve our model's performance. And then finally, I'll sort of conclude and talk about some future work. Yeah, so one of the main sort of stages of drug discovery where you want to have a good binding affinity prediction model is the hit identification stage, which is all about finding, a set of molecules that bind to your target of interest. And a commonly used computational method here is structure-based virtual screening. So for a given target, we search through a large library of compounds, and for each compound, you predict whether or not your compound binds to your target of interest. And these libraries can be really large, can have up to a few billions of molecules in them. So for a subset of your library, you can use docking tools to predict how this molecule might bind to your protein. And finally, you can use a scoring function to predict how tightly that molecule binds. So yeah, that's called binding affinity prediction. And some of the most important aspects of your binding affinity prediction model in this scenario is that it needs to be fast, it needs to be high throughput. We're working with a very diverse set of molecules here, so it needs to be able to rank a very diverse set of molecules, and it needs to be able to generalize to some extent, meaning that the model has to be able to perform well on molecules and proteins that are sort of dissimilar from what the model has seen before during training. The other scenario I want to talk about where binding affinity prediction is used is lead optimization. And at this stage in the process, we hope to have found, a set of molecules that bind to our target. And this stage is more about refining those molecules to optimize for many different properties, but binding affinity being one of them. So we want to make sort of small changes to our molecules that we've already found to increase their binding affinity. And a standard sort of computational workflow here is to generate a congeneric series of molecules. So a set of molecules that share the same sort of core structure and differ only in some functional group, dock them all into the same protein pocket and predict and rank their binding affinity, usually using a set of physics-based models called FEP. And these models are quite computationally expensive and usually not open source, but they're the best sort of we have and can be quite accurate. So in contrast to the hit identification stage I talked about, where we have a very diverse set of molecules, here we're looking at sort of very small differences. So we're looking for different things in our model. We want the model to be able to rank a series of very similar molecules. We still want our model to be fast, but it's not as important as in the hit identification stage, and generalizability is always important. So the main difference lies in the sort of level of diversity in the molecules we're trying to rank. With those sort of two different stages in mind, I want to talk about how machine learning scoring functions are usually trained and tested in literature. So the most commonly used training data is PDB bind, and the 2020 version of this data set has around 20,000 crystal structures of bound protein ligand complexes, along with binding affinity measurements. And a subset of this data set is called CASF 2016, which has 285 complexes. And this is by far the most sort of commonly used benchmark out there. It was developed as part of the sort of comparative assessment of scoring functions. And it was created by clustering data points in PDB bind by sequence identity, so by protein similarity. And then from each cluster, 5 complexes were selected into a test set. And because it was generated this way, CASF 2016 is very much an in-distribution benchmark, meaning that the sort of complexes in the test data are highly similar to what you would find in the training data set. And over the past sort of decade, dozens of different models have been published with various different architectures and sort of featureizations of the protein ligand complex. And all of them have achieved a Pearson correlation ranking ability of around 0.8 on this benchmark, with sort of very limited improvements over time. And so, but there are quite a few sort of aspects of binding affinity prediction which are not measured or estimated using this CASF 2016 benchmark. For example, we don't get an idea for how well our model generalizes. The test set consists of sort of various different targets and various different sort of sources of binding affinity data. So a part of the performance you can get on this benchmark comes from simply being able to sort of predict the average binding affinity of certain targets or average binding affinity of certain molecules. This has been shown by sort of training models that learn only from ligand properties or only from protein properties, and they still achieve this sort of 0.8 correlation on this benchmark. And finally, this benchmark doesn't really estimate performance for lead optimization. So we set out to answer sort of two main questions, like can we develop a benchmark that better estimates generalizability of machine learning scoring functions, and how applicable are these scoring functions for lead optimization? Yeah, so I want to talk about our new benchmark called OOD test, which was created to sort of try to get a better estimation of generalizability. It's quite a simple idea. It's a new dataset split of PDB bind 2020, and we created it by sort of minimizing the maximum similarity between training and test set complexes, both in terms of ligand and protein similarity. And we do this by using hierarchical clustering, using single linkage. And we use like a distance metric that takes into account the Tanimoto similarity of the ligands and the sequence identity of the proteins. And what we end up with is a subset of around 300 complexes. And if you take any complex from the test data and any from the training data, then the ligand similarity will be less than 0.5 in terms of Tanimoto similarity, and the protein similarity will be less than 50% in terms of sequence identity. Yeah, to get a better estimate for performance for lead optimization, we use this FEP benchmark. And rarely when you sort of see a new binding affinity prediction model published is it compared against these physics-based methods called FEP, although they're used very widely in industry. And so the nice thing about this benchmark is that it contains a lot of data, but it contains experimentally determined binding affinity, but also predictions from FEP+, which is Schrodinger's FEP model. So it contains 43 congeneric series of ligands, each series having 10 or more ligands. So binding to various different targets, but within each series, they're binding in the same protein pocket. And typically within each series, they have quite a narrow binding affinity range, which makes it a very challenging benchmark for models to sort of rank the binding affinity within each series. Yeah, so before I show you some results on the benchmarks that I've mentioned, I want to briefly talk about our new model called AEV-PLIG. The main idea with AEV-PLIG was to use sort of recent improvements in geometric deep learning, coupled with a sort of rich representation of protein-ligand interactions, while still keeping the model quite simple. So AEV-PLIG is a GNN-based model. It takes in a graph that has the topology of the ligand, and then we encode the interactions with the protein through sort of the feature vectors of that graph using atomic environment vectors. So for each atom in the ligand, we sort of summarize the protein atoms that surround it using Gaussian functions. And each entry in the atomic environment vector is sort of only focuses on certain protein atom types and only a certain distance range away from this reference ligand atom. Then this input graph is propagated through 5 GATV2 layers, which is a highly expressive sort of attention-based GNN layer, followed by a global pooling method, and then an MLP. So here are some initial results of our model on these the two different benchmarks I've talked about, CASAP 2016 and our new out-of-distribution test. And we do see a drop-off in performance as expected, since our new benchmark is more challenging. It seems to be particularly challenging for the model to sort of rank these tight binders here on the left. We also sort of tested various different machine learning models out there on the same benchmarks, and a similar story can be told about those. We see a significant drop-off in performance when we move from CASAP 2016 down to OOD test. We see a smaller drop-off for AEV-PLIG compared to most of these other models, which we find encouraging. But some of the main, or one of the most sort of interesting results from this table here is the performance on the FEP benchmark. So we measure performance on the FEP benchmark using the mean Pearson correlation across the 43 different series that I talked about, weighted by the number of ligands in each series. And our model performs the best and achieves a mean correlation of 0.4, which is nowhere even close to the performance we get using FEP+, which is closer to 2.7. So all of these models that we tested are not really applicable for lead optimization as it stands, sort of out of the box, trained only on PDB bind. So we wanted to sort of try to improve the applicability of our model for lead optimization. And we tried this by using semi-synthetic data. And what I mean by semi-synthetic is that the binding affinity is still measured in a lab, but the structural data is modeled with constrained docking, or a version of constrained docking. And so we have this data set called BindingNet, which achieved this by sort of scraping Kemble for binding affinity data, specifically where the binding affinity data comes from the same protein and a very similar ligand. So then they can use this sort of reference crystal structure from PDB bind to sort of guess or predict how this similar ligand might bind into the same pocket. And they follow a few steps to sort of model this structure. They align the reference ligand or the new ligand to the reference ligand using maximum common substructure. And then they generate conformers and minimize with a force field. And finally, they rank all the conformers with MMGPSA and filter the best one. So they were able to generate around 70,000 complexes this way. And Binding DB also has a similar dataset of 9,000 complexes, which we also used. And we do expect this data to help for lead optimization, not only because it's sort of expanding the amount of data we use for training, but also it's increasing the amount of times we have a set of similar molecules binding to the same protein. And so the model should be able to learn from these really similar sort of complex structures and how that relates to differences in binding affinity. And this is exactly what we find. We find that adding or retraining AAV-PLIG with the semi-synthetic data greatly improves our performance on the FEP benchmark. Here we have sort of AAV-PLIG trained only on PDB bind in green. And in blue, we have AAV-PLIG trained on both PDB bind and the semi-synthetic data. And in red, we have FEP plus for comparison. And the x-axis shows the maximum Tanimoto similarity between the ligands in the training data and the ligands in the FEP benchmark. So we're being more strict with sort of dataset leakage as we move from left to right. And like I say, we see a big improvement in performance, sort of taking our model from not really being sort of applicable for lead optimization to being, still slightly worse than FEP plus, but comparable. Especially if you take into account that AAV-PLIG is around 400,000 times faster than FEP plus and is open source. When it comes to the other benchmarks I've mentioned, we don't see any significant difference in performance when adding the semi-synthetic data. We don't know exactly why that is, but it could be because we're still restricting ourselves to PDB bind, meaning although we're sort of adding more data, we're not adding data in new areas in chemical space. So that could be the reason. Another interesting and exciting result we found is when we sort of gradually train with more and more of the semi-synthetic data. So starting from the left here, we're only training on PDB bind, and then we're gradually training with a higher and higher proportion of the semi-synthetic data set. And we see a gradual increase in performance on the FEP benchmark. What we don't see is a spike in performance, which then tapers off. So this result might indicate that if we put some more effort into generating more of this data, then we might see even further improvements, which would be really good, but that remains to be seen. So with that, I'd like to sort of conclude. FEP plus is still sort of better than the machine learning methods we tried out, but the gap is getting smaller. Especially, we believe AAV-PLIG is already quite useful when you think that when you note that it's 400,000 times faster than FEP plus and is open source. We believe these methods might work quite nicely in tandem as well. So you could sort of explore a bigger series or set of ligands and then use AAV-PLIG to predict and funnel the best guesses into FEP plus or another FEP method in an active learning scenario, for example. FEP also has to have quite similar ligands so that it's able to do relative binding free energy calculations, whereas AAV-PLIG doesn't have that restriction. So you might be able to explore a sort of more diverse set of ligands in your lead optimization strategy using a machine learning model. Something we want to address for future work is that AAV-PLIG has never seen sort of negative examples. It's never seen a bad binding pose, or at least not a very bad binding pose, or it's never seen a non-binder. So we don't expect it to work for virtual screening and docking rescoring. So we plan on sort of retraining AAV-PLIG using negative examples generated through, for example, decoys and redocking and things like that. So thank you for listening. Special thanks to my supervisors, Garrett Morris and Charlotte Dean, and of course my group, OPIC. I'll take any questions if you have any.
Thank you, Isak. Yeah, we'll take some questions from the room first. Do you have microphones again? Any questions from anyone?
Thank you. How long does it take to train your model, and what kind of resources do you need for that?
So it takes around 24 hours to train our model on PDB bind and the semi-synthetic data on sort of pretty standard GPU. Yeah. What was the other question?
You've answered the second part, so yeah, on standard GPU. Okay, thank you.
Are there any more questions from the room? Otherwise, we'll go to the ones online. If somebody could help me, what the first one will be. What was it? This is all from
Morgan's is the first one. There are two questions right now, currently.
Lower, no. No. Well, here, this one. Morgan Thomas?
Yes.
Excellent. So, nice work. Did you explore methods to estimate the applicability domain of AEV-BLIC so that a user might know when to have more or less confidence in the model?
Not really. I mean, we did the experiment where we sort of gradually removed more and more similarity from the training data, and it still holds up decently well. But we haven't developed sort of a confidence estimation for our model yet, which would be actually sort of a nice direction for future work. So currently there's probably a bit of uncertainty for how far you can push the model before it won't sort of do very well anymore.
The question from Serhi, on your preprint, you also showed that addition of target-specific 3D structures further boosts the model, and our in-house results support this finding, even when target-specific docking-derived 3D data is used. I'm wondering if you have evaluated the optimal number of complexes to add to get the best effect on prediction accuracy.
This is actually a very good question, because we did have a different benchmark that we used for our preprint. And we found that sort of fine-tuning our model with a few complexes from our congeneric series of interest helped quite a bit, which we no longer found on the new sort of bigger benchmark. And we don't fully know why that is. It's actually quite surprising to me in general that only a few complexes in general would work for fine-tuning, because it's such a small set of examples you have. So my expected sort of intuition with that fine-tuning wouldn't work. Maybe it helps because the model sort of has seen slightly different structures during training, meaning the structures have been generated in a slightly different way during training than during testing. And so the model learns to sort of adapt to that. But no, I don't know what the optimal number of fine-tuning data points is, because we no longer see that improvement.
Okay, and the question from Kepa, does your model work for neutral and charged species and ligands, or do they perform differently?
So currently we don't have any clear-cut scenario where AV performs badly or performs well. We've been trying to figure out sort of a relatively simple explanation for when you might want to use it, when you might not want to use it. And currently we don't have a clear sort of easy answer for it. So we have found scenarios where it works for neutral molecules and charged, and scenarios where it doesn't work as well. So yeah.
And then a further question on the management of the sort of risk of overfitting while training with the augmented data. How do you manage that risk?
We don't manage it. Well, we manage it through early stopping during training, and then we sort of make sure that it isn't overfitting during testing by sort of training on different levels of similarity to the test data.
Okay, we should check. Are there any further questions from the room before we finish off on the online questions? Anyone else wants to ask Isak something? If If not, afterwards, more chances in the pub. Actually, not for Isak because he's online, but we're going to the pub. Well, maybe a couple more questions. Have you taken a look at Plinder? It's allegedly a much larger, high-quality PLI data set. They also have leaderboards, etc. Question.
Yeah, I know about Plinder. I haven't trained AV-Plick on Plinder yet. And I don't think there's like binding affinity data for all of those data points, but definitely some proportion of it. So retraining AV-Plick would be interesting on Plinder for sure.
Let's move to the next question from Priya. The LIT PCBA dataset is claimed to be unbiased set, an unbiased set for ML training. Have you tried that one?
No, I have not tried that one, is the short answer.
Okay. Well, no is a complete sentence.
Yeah.
Thank you. And then we have a word of caution from Francois. I don't think that's a question. It's just a comment. So you can read it. Well, if there's no further questions, then again, let's thank Isak for his presentations today. Matter of fact, let's thank all the speakers once more because, brilliant presentation.